Students, now we will discuss about amino acids. In my previous video, we saw that amino acids are micromolecules. Their molecular weight is less than 800 Dalton and they are found in acid-soluble pool. Now just see here what are these amino acids. Amino acid contains the amino group and carboxylic group. These two groups are attached to the alpha carbon atom, so they are also known as alpha amino acid. Just to see here, this is the structure of an amino acid. So now here we can see that this is our alpha carbon atom. Now to this is attached the carboxylic group and the amino group. So this amino acid will be known as alpha amino acid. So in amino acid we can see there is a carboxylic group and a amino group. Clear? Now we can say that they are substituted methane. In our previous classes we have studied about the structure of methane, CH4. Now what happens here, this H is replaced by carboxylic group and this hydrogen atom is replaced by the amino group. So we can say that amino acids are substituted methanes. Clear? Now here I have drawn 3 amino acids which are given in your NCERT, and you have to remember the structure of these 3 amino acids. The first one is glycine. Here you can see this is the alpha carbon, and to this alpha carbon is attached the carboxylic group, the amino group. Here you can see the hydrogen atom, and here you can see the variable group, and here in this case the variable group is H, that is hydrogen atom. Next is alanine. In the case of alanine we can see that the R, that variable group, is actually the methyl group, and in serine it is CH2OH. So you have to remember the structure of these 3 amino acids as it is given in your NCERT. Just see again, glycine. Here we can see that R is equivalent to H, or we can say that that variable group here is H. In the case of alanine it is CH3, and in the case of serine it is CH2OH. Clear? This you have to remember. Next, it is amphoteric compound. Now why it is called amphoteric compound? Because it contains a carboxylic group and an amino group. So that's why it is called as an amphoteric compound. Now what happens if you keep this amino acid in acidic pH? Clear? Suppose we have kept this amino acid in acidic pH, what will happen? We know that acid, they releases the hydrogen ion, and that hydrogen ion will be accepted by the amino group and it will become NH3 positive. Clear? If you keep this amino acid in acidic pH, what will happen? It will take, the amino group will take hydrogen atom and it will get the positive charge. Now if you keep this amino acid in alkaline pH, first we have kept it in acidic pH. Now I am telling that if we kept it in an alkaline pH, now what will happen? This carboxylic acid, it will release the hydrogen ion and it will be accepted by this alkaline medium and it will become negatively charged. Clear? But what happens if we maintain a pH where there will be only intramolecular transfer of the hydrogen ion? Suppose we have maintained a pH where what will happen? This hydrogen ion will be accepted by this amino group. So that pH where there is intramolecular transfer of the hydrogen ion, that pH will be called as isoelectric point. Just remember that pH where there is intramolecular transfer of hydrogen ion, it will be known as isoelectric point. It means the hydrogen ion which is given by COOH group, it will be taken by the amino group. That pH is known as isoelectric point. Clear? At that point, what happens? The Zwitter ion formation takes place. So at isoelectric point, amino acid is present in the form of Zwitter ion. Clear? So there, what happens when it becomes positively charged? It will move towards the negative electrode. When the amino acid becomes negatively charged, it will move towards the positive electrode. But here there is no movement, or we can say the neutral amino acid, what they are in the form of Zwitter ion. Clear? So what is isoelectric point? It is that pH where there is intramolecular transfer of the hydrogen ion from the carboxylic group to the amino group. Clear? And that point is isoelectric point, and amino acid at that point is known as Zwitter ion. Now we know that there are 300 types of amino acid, but actually 20 amino acids are required for the protein synthesis. Now out of these 20 amino acids, the 10 amino acids are essential. Now what does essential amino acid means? It means we have to take them in our diet. Clear? Now those which are being made in our body, that will be called as non-essential amino acid. But those which are not being made in our body and which we have to take from outside, either from by plant, it will be called as essential amino acid. So there are 10 essential amino acids, and we have to remember the name of these 10 essential amino acids. So what are those 10 essential amino acids? For this there is a mnemonic, and if you remember this mnemonic, you will never forget the name of the 10 essential amino acids. Now what are these 10 essential amino acids? To remember it, you have to remember this mnemonic. Try this VIP mol. The mnemonic is try this VIP mol. You will remember it. Now try. This try stands for one amino acid which is essential, that is tryptophan. Just remember, try stands for tryptophan. Second, this. In this we will take two amino acids. This TH stands for one amino acid, that is threonine, and HIS, it stands for histidine. Clear? Now next is VIP. V stands for valine, I stands for isoleucine, and P stands for phenylalanine. Okay. Now mol. In mol, M stands for methionine, L stands for lysine, and another L stands for leucine. So here we can see 1, 2, 3, 4, 5, 6, 7, 8, 9, and there are 10 amino acids. Clear? So by remembering this mnemonic, we can remember the 10 essential amino acids. Now these amino acids are joined with the help of peptide bond. Now what happens? These amino acids are joined together to form a polypeptide chain. Clear? Now in between this amino acid, there is a bond, and that bond is known as peptide bond. So just see here, this is the first amino acid. Here we can see the alpha carbon, the carboxylic group, the amino group, hydrogen, and the variable group. This is the first amino acid. This is the second amino acid, where we can see the alpha group, the carboxylic acid, and the amino group. Now during the peptide bond formation, the OH from COOH of the first amino acid and the H of amino group, they will be loosed in the form of water. And there will be the formation of the peptide bond. So there is the formation of peptide bond between two amino acids, and there will be the release of H2O. Clear? So the amino acids are joined with the help of peptide bond to form proteins. So it is very important question, what type of bond is present there. So you have to remember, it is the peptide bond. Clear? So like this, a very long chain will be formed of amino acid, that will be known as polypeptide chain. In polypeptide chain, this terminal will be known as N-terminal, and the last one will be known as the C-terminal. Clear? Now next we will see the classification of amino acid. We can classify the amino acid as acidic amino acid, alkaline amino acid and the neutral amino acid. Now in acidic amino acid, if there is one amino group, clear, there is one amino group and two carboxyl group, so that amino acid will be called as acidic amino acid, and it will have net charge negative because it have two carboxyl group. Clear? So in acidic amino acid we have one amino group, two carboxylic group, and it will have a net charge negative, so it will be called as acidic amino acid. Two example we have to remember. There are only two amino acid out of 20 which are acidic amino acid, that is glutamic acid and aspartic acid. Clear? We have seen that acidic amino acid have one amino group, two carboxylic group, so they have net charge negative, and the two examples are glutamic acid and aspartic acid. Now the second one is the alkaline amino acid. So here what we will have? We will have two amino group and one carboxylic group. Here we can see alkaline amino acid. It has two amino group and one carboxylic group. So the net charge will be positive, and there are three amino acids which come in this category, that is histidine, arginine and lysine. Clear? And the third one is the neutral amino acid. Here we will have only one amino group and one carboxylic group. And out of the 20, we have seen 2 here acidic, 3 here alkaline. So out of 25 have gone, so we will have rest 15 as neutral amino acid. This amino acid does not move in electric field and they are in the form of Zwitter ion, these neutral amino acid. Clear? Since they are negatively charged, so they will move towards the anode. These are positively charged, they will move towards the cathode. And these are neutral, so they will not move in the electric field and they are present in the form of Zwitter ion. So this was all about the amino acid. Thank you.